Hello friends, welcome to the Biology Forum. We are going to start with the second part of our lecture series on the circulatory system. In this lecture series, these videos, we are going to discuss about blood and lymph. So let's start. When we talk of the circulating fluid, that is our blood, it is a special connective tissue. We could also call it as the fluid connective tissue of our body. It is made up of two components. There is a fluid matrix, the fluid part of the blood, which we call as plasma. If you can see, this is the plasma. And you have the formed elements or the cellular components of the blood. So altogether, when we talk of the blood, there will be the fluid part and the formed elements. So let us talk about the plasma, which is the fluid matrix of the blood. It is a straw-colored viscous fluid which constitutes about 55% of the blood. That means 55% of the blood is made up of plasma, whereas 45% is made up of the formed elements or your cellular components. Plasma by itself is made up of 90 to 92% water and 8 to 10% of dissolved components. Now, what are the dissolved substances that are flowing in the plasma? These are around 6 to 8% of proteins. These include even fibrinogen, which helps in the clotting or coagulation, globulins, these are also proteins. You have minerals in the form of ions like sodium, calcium, magnesium ions. You have glucose, which also needs to reach to each and every cell in the dissolved form. We have the lipids, we have wastes like urea, and even the chemical messengers from our endocrine glands, the hormones. All these are transported in the dissolved form through the plasma. What about the formed elements? The formed elements constitute 45% of the blood, and they're made up of three major types of cells. The first are the erythrocyte, also called as the red blood corpuscles. Erythro means red and cytes are cells. So these are the red cells or the red blood cells. Leukocytes, leuko means white, white cells or the white blood corpuscles. And thrombocytes, that is the platelets. Now let us talk about the erythrocytes. The erythrocytes, if you can see in the figure, they are the most abundant of all the cells. A healthy adult man has around 5 to 5.5 million cells per cubic millimeter of blood. The red color of blood is because of a red colored iron containing protein called as hemoglobin, which imparts a red color. So this hemoglobin pigment is present inside the red blood cells, hence the color red to the cells as well as to the blood. The healthy individual, the amount of hemoglobin that is present is around 12 to 16 grams per 100 ml of blood. The shape of the erythrocytes, they are all biconcave disc-shaped cells. They are devoid of nucleus. When they are produced initially, they do contain the nucleus, but as they start maturing, this nucleus disappears. They are produced in the bone marrow and their lifespan is around 120 days. So that means they remain in circulation for about 4 months, after which they are removed. They are generally destroyed or removed in the spleen. The leukocytes, on the other hand, they are, as the name suggests, leuko means white, so they are colorless, but they are nucleated. They are less in number as compared to your erythrocytes, around 6,000 to 8,000 per cubic millimeter of blood. The lifespan of leukocytes is also very less, it's for a few weeks. Now, when we talk of leukocytes, there are two main types of leukocytes or the white blood corpuscles, granulocytes and agranulocytes. The simple term means that the granulocytes will show granules in their cytoplasm, whereas agranulocytes are devoid of any granules. Of these, the granulocytes are of three types: neutrophils, eosinophils, and basophils. whereas agranulocytes are of two types, monocytes and lymphocytes. Let's talk about the granulocytes. The neutrophils, basically they get stained with neutral dyes. They have around 2 to 5 lobed nucleus. They constitute the major part of WBCs, that is around 60 to 65% of the total WBCs are made up of the neutrophils. They are also called as the polymorphonuclear leukocytes or polymorphonuclear cells. The main function is phagocytosis, that is they move by amoeboid motion. Whenever there is any foreign body or a pathogen, they move towards it and engulf it to destroy it. The eosinophils, as the name suggests, they are also called as acidophils because they pick up acidic dyes like eosin, hence the name eosinophils. They constitute around 2 to 3% of the total WBCs. They are generally associated with allergic reactions. So when any allergic reaction sets in, the number of eosinophil count increases more than what is a normal, and at such times we call it as an eosinophilia. Basophils, on the other hand, get stained with basic dyes like methylene blue. They constitute a very small portion of the WBCs, around 0.5 to 1% of the total WBCs. So in a smear, generally, you will not get to see basophils at all. They secrete histamine, serotonin, heparin, and are also involved in inflammatory reactions. These things, you have to remember these points, these functions, because they can be asked in your MCQs. Now let's talk about the agranulocytes. In that we had already discussed there are two types of agranulocytes, the monocytes and the lymphocytes. Monocytes constitute about 6 to 8% of the total WBC count. They are also phagocytic in function, similar to the neutrophils. The lymphocytes are the next highest in number, around 20 to 25% after the neutrophils. They are of two types. You have the B lymphocytes and the T lymphocytes, and these are responsible for immune responses in our body. So they are very, very important for the immunity that we develop against certain diseases. Yes, the lymphocytes play a major role in this. Right, this is how the various white blood cells look. This is a neutrophil. If you can see the number of lobes, it's almost 2 to 5 lobes. There are 4 lobes can be seen here. This is an eosinophil. It is colored with an acidic dye, so it appears reddish. This is a basophil stained with a basic dye. So if you can see, there is a bluish tinge, a purplish tinge, monocyte and lymphocyte. You can see the kidney-shaped nucleus here in the monocyte and the lymphocyte, which helps in immunity, in our immune responses. Yes. We come to the third type of the cellular component, that is thrombocytes. When I talk of thrombocytes, that is, they are the cells, they are platelets, the cells which help in thrombus formation, that is clot. A clot is known as thrombus. So these platelets or thrombocytes play a major role in the process of coagulation or clotting of blood. Hence, they are very, very important. These are cell fragments. They are very small cells which are produced from the megakaryocytes. What are the megakaryocytes? These are special cells present in the bone marrow, and from there, these are the fragments of these megakaryocytes, which we call as platelets. The normal range of platelets is around 1,50,000 to 3,50,000 per cubic mm of blood. So if you really compare, the maximum number of cells are going to be the RBCs, followed by the platelets, and lastly the WBCs. The important factor, the function of thrombocytes, is they play a major role in clotting of blood, also called as coagulation of blood. So let's learn what is coagulation of blood. Now, this is a very important protective mechanism that our body has. Whenever there is an injury, you might have noticed that there will be bleeding, and after some amount of time, 2 to 3 minutes, this bleeding stops. This bleeding stops because there is a formation of a plug or a clot at that site of injury, and this prevents further blood loss. That is why this is a very protective mechanism for us, because of this coagulation or clotting mechanism, we are stopped from losing a lot of blood. So it is very protective in function. So how do the platelets help in this function? Whenever there is an injury at the site, so there will be a break in the continuity of the blood vessel, as you can see here, right? Because of this injury, blood starts flowing out from here. Now immediately when there is an injury or the break in a continuity of the blood vessel, the platelets are stimulated to release certain factors which we call as the clotting factors. Now these will help in the release of an enzyme, the thrombokinase. This thrombokinase enzyme, along with the presence of calcium ions which are already in the plasma, will act on the inactive factor called as prothrombin, which is in the blood, and will convert it into active thrombin. Now this active thrombin, along with the presence of calcium ions, will act on the soluble fibrinogen. If you remember, I had spoken about fibrinogen being dissolved in the plasma, so it's in the soluble form. The thrombin will act on this fibrinogen, which is in the soluble form, and will convert it into insoluble fibrin. When we talk of fibrin, we have to understand that there will be fibers in this. So a meshwork, these fibers will go and form a meshwork or a network at the site of the injury. What happens is, as the blood is flowing through this blood vessel, the dead elements and some of the cells will get trapped in this meshwork. And soon what will happen is it will contract and will plug this site of injury. It's like as if that open area is plugged, and this is called as the clot or a thrombus. So this is how our platelets help us in they prevent excessive loss of blood through this process of coagulation by forming the thrombus. That is why platelets are also called as thrombocytes. Right. We come to the next part where we have always know about our blood group system. It is having two components, the ABO system and the RH system. So whenever anybody talks about their blood group, they will always talk of, for example, if my blood group is BRH positive. So I have one component which is the ABO system and the other is the RH system. The system of blood groups was first introduced by Karl Landsteiner, and what you have to understand is the presence of certain antigens on the surface of the RBCs is what constitutes our blood group. So what does it mean? If my blood group is A, on the surface of the RBCs there will be the presence of antigen A. So in the serum or in the fluid part, you will be having antibodies against the other one, that is B. So I will be having anti-B antibodies in my circulation. So on the surface of the RBC, I am having the antigen A, if you can see this. Similarly, the B blood group will naturally have antigen B on the surface of the RBCs, whereas in the circulation there will be anti-A antibodies. So simple, if I'm having my blood group as AB, it will have both A and B antigens on its surface. So there will be no antibodies in circulation. That is why this is a very important point that you have to remember when we talk about universal donors and universal recipient blood groups. When I talk of a O blood group, that means there is no antigen on its surface. It's O. So no antigens on its surface, but in the circulation you will have anti-A and anti-B antibodies. This is the basic concept of the four blood groups from the ABO system. So because of this, simple, why we need to do cross-matching is that if I'm having my blood group is B, that means I'm having B antigen on the surface of my RBCs and anti-A antibodies in circulation. That is why if A blood group is given to me, what will happen is these antibodies, anti-A antibodies, will destroy it. Similarly, what happens is for an AB blood group, because they are having both the antigens on the surface but there are no antibodies in circulation, they can receive blood both from A, B as well as O. So blood group AB becomes a universal recipient. A recipient is one who can receive the blood. So they can, an AB blood group A person with an AB blood group can receive blood both from A, B as well as O. Now why O? Because on the surface of O there are no antigens at all. So you can very easily give O blood group. So blood group O becomes the universal donor because they have no antigens on their surface, so they can give to A, B as well as AB. So question asked most of the times is the universal donor blood group is O, universal recipient blood group is AB. This is one system, the ABO system. Now if you talk of the other system, the other component is the RH. RH stands for the rhesus factor or the rhesus antigen. This was first named because the same factor was also found in the blood of the rhesus strain of monkeys. So from that, because even we have the same factor, it is called as the rhesus factor. So if the rhesus antigen is present, we call that particular blood group as Rh positive or rhesus factor positive. If the Rh antigen is absent, we call it as Rh negative. So if my blood group is BRH positive, it means that on the surface of my RBCs are present the B antigens. In circulation, you have the anti-A antibodies circulating, and since I'm Rh positive, that means the Rh antigen is also present on the RBCs. This is how we understand. So let us see, we also have Rh positive and Rh negative. So just let us see in a nutshell whom they can donate to and whom they can take from. If it is an O negative blood group, they can donate to all the blood groups, but they can take only from O negative because they're not having the Rh antigen in them. So they can take it receive only from O negative. O positive can donate to all the blood groups. AB positive, A positive, B positive and O positive can take from O negative and O positive. You have to remember that even if the O blood group is a universal donor, they can receive only from O group. Right. Then we have A negative. If it's A negative, they can give to AB negative, AB positive, A positive, A negative can receive from O negative and A negative only because theirs is a negative, Rh negative. If it is A positive blood group, they can donate to AB positive and A positive, and they can take from O negative, O positive, A negative and A positive. B negative can donate to B negative, B positive, AB negative and AB positive. O negative, they can take from O negative and B negative only, not from the positive. The B positive can donate to B positive blood group, and AB positive blood group can take from O negative, O positive, B negative and B positive. AB, now we are talking of the universal recipient. So AB negative and AB positive, because it is negative, they can donate to AB. They are recipients, but they can give to only AB blood group. So they will be donating to only AB negative and AB positive, but they can take from O negative, A positive, B positive, B negative also. Whereas here, if it is AB positive, they can give to only AB positive, but they can receive from all. It's a universal recipient. On top of it, it is a positive blood group. So they can Rh positive. So they can take from all the blood groups. So this is the best blood group possible. If you're going to have that, it's a very good blood group to have. Right. So once we have learnt about one of the circulating fluids, that is blood, we are going to talk about lymph, which is also called as the tissue fluid. Now what exactly is the lymph? As the blood passes through the capillaries, now it is flowing with a lot of pressure. The capillaries have a single cell wall thickness called the endothelium. So this pressure, because of this pressure, some amount of water, the fluid portion, along with some small water-soluble substances, not the larger proteins. The larger proteins will not be able to squeeze through the capillary walls. The small water-soluble substances, along with the water, will move out into the spaces between the cells. That is why the lymph is also called the interstitial fluid or the tissue fluid. Because what happens is it surrounds the cells and it bathes the cells with it. So as the blood is flowing, because it is flowing with a lot of force, the heart is pumping it with force. through the capillaries as it flows because of the pressure, some amount of fluid along with the small water-soluble substances will squeeze out into the intercellular spaces, which is also called as the lymph or the interstitial fluid. Along with this, WBCs too can move out of the capillaries into this fluid, but no RBCs and platelets will be seen. What happens because of this tissue fluid? Exchange of nutrients and gases between the blood and the cell occurs through this fluid. So what happens is, because the cells are completely surrounded with the lymph or the interstitial fluid, exchange of gases takes place through this. They also contain lymphocytes. So what is the lymphatic system? It is an elaborate network of vessels which collects the lymph and drains it back to the major veins. It's the lymphatic system. Because this fluid which is around the cells needs to also go back into the circulation. It cannot remain there, because then the blood will lose a large amount of water and the viscosity of blood, the pH of blood will be lost. So we cannot afford that to happen. So the system is such that the lymph which is around the cells is drained back, is collected by the lymphatics or the lymphatic vessels, and they are drained back into the veins, which is then carried to the heart and back into the circulation. That is the lymphatic system. It is an elaborate network. The lymph is carried through lymphatic capillaries, just like you have capillaries at the end of the arterioles. Similarly, we'll have these fine vessels called the lymphatic capillaries, which will then take this lymphatic fluid to the lymphatic ducts, and from there to the veins. The veins will then carry it to the heart. As it contains specialized lymphocytes, as I told you, WBCs too can move through amoeboid movement, and they can squeeze out of the capillaries. So the lymphocytes, the lymph is abundant in these lymphocytes. So the lymph plays a major role in our immune responses. This is how the lymph is trained. So if you can see, through these capillaries, you have the lymphatic capillaries too. This is a secondary network that you can see. All the capillaries are draining here. You have the lymph node here. You have the ducts out here at regular intervals. You have larger lymphatic vessels, and if you can see, ultimately it is draining into the vein. It can be near the junction of the subclavian vein and the internal jugular vein. Ultimately, it goes into the heart and back into the circulation, and this continues. So this is an alternative system that is present along with the regular circulation of blood. So this is our lymphatic system. So this concludes our session or lecture series, the second part on circulatory system, where we have discussed about the circulating fluid, that is blood and lymph. Keep watching if you want to know more about our blood vessels, the channels through which these fluids are flowing, that is our arteries, veins and capillaries. Thank you.